So let's start. Yesterday we done about T cell selection, okay, and also one topic regarding T cell activation. Let's we continue. Why we need selection? Why we need selection? Because the some cells we need some gene arrangement, okay. Like we need to teach, or there must be some foreign antigen that the cell can be active against them, and they not have like no binding or attraction for normal body cells, okay. They no need to react against our body protein. If you not do this selection, otherwise the what happens, their cells, they can start, they are self-reactive. They're just self-reactive, and they can stimulate autoimmune diseases, okay. They can stimulate autoimmune diseases. Clear? It's the importance of selection. What's the role of thymus in T cell? Role of thymus. Cells coming from bone marrow, they are immature, okay. Now when they enter to thymus, in thymus the T cell now become mature. Thymus become, in the thymus they become mature, okay. What's the meaning of maturation? I already explained. The step, thymic education, okay. This is a maturation inside the thymus. It's a role of thymus. Clear? It's the role of thymus. When the cell become mature, immature T cell enter to thymus. Now when enter to thymus in the cortex, then the thymus medulla, then leave the thymus as a mature cell. Mature cell mean well educated cell. Have ability to differentiate between the difference between self protein and non-self protein, okay. Non-self protein, non-self antigens come from outside. It's the role of thymus. The development of T cell and selection. I already explained about the selection, okay. Negative selection and positive selection. So for negative selection, so that cell must die. Understand? Because that cell not follow. So when the cell enter the thymus, if the cells do not like follow the rules and regulation of thymus, then what the education given by the thymus, they are negatively selected, okay. They are negatively selected. The positive selected cell, they are positively selected cell, they must enter our defense system. They play a key role in our defense system, okay. So it's a negative selection and positive selection. This education we also done negative selection, positive selection, okay. Important surface marker yesterday we also done. The types of T cell, regulatory T cell, Th1 cell, Th2 cell, and suppressor T cell. Th1 cell for cell mediated immunity, okay, for cell mediated immunity, and Th2 cell for antibody mediated immunity. When the job is done, suppressor T cells suppress these cells, okay. Regulatory T cell regulate all T cells, regulation. Antibody production done by B cell change to plasma cell produce these 5 types of antibody, okay. This activation we also done, the CD4 cell change to Th2 and Th1. They produce some cytokines, interleukin 4, 5, interleukin 2. They activate macrophage and CD8 cell. So for killing, we use cells. When they stimulate B cell, the B cell produce antibody, okay. Still TH1, this already done. TH1 and TH2, what they're doing, okay? This job is clear about TH1, TH2, everyone clear? Last lecture.
Yes.
Okay, this
Yes.
activation we also done primary signal and secondary signal, okay? Must keep in mind, what's a primary signal? Primary signal, MHC protein interact with T cell receptor, okay? MSC 1 for which receptor cell? Which cell? 1 for CD8, okay? And if the protein is MSC2, then T cell receptor of CD4, okay? This is a primary signal. The secondary signal between B7 and CD28, okay? Another secondary signal, CD40, CD40 and CD40L. These are two
L.
secondary signal, okay? And this secondary signal for macrophage, secondary signal for dendritic cell. They use this.
Dendritic cell.
If the B cell give antigen to T cell, then secondary signal not this, okay? Then secondary signal CD40 and CD40L. Okay, so we already done, okay? Secondary signal. Okay, let's see what is energy. It's not energy, okay? It's a energy. It's not energy. Energy is activation. Energy means no activation, no energy. Oh, no energy. And A means without. What is this condition? What is the situation here? Co-stimulatory signal, the secondary signal there, yeah? There's a co-stimulatory signal, secondary signal. What was the secondary signal? B27 with CD28. If this signal not working, like if this signal is not working, what happens? If this signal not working, the cell become active? No.
No.
T cell not fully active, okay? T cell not fully active, understand? T cell not fully active, clear? T cell not fully active. So if we not have this signal, our T cell not become full active. This situation known as energy, okay? Antigen, you see, antigen enter your body and macrophage and dendritic cell give this antigen to T cell, okay? The primary signal is there. This is a primary signal, MSC2 or a T cell receptor. This is a primary signal. Primary signal is not sufficient to activate for full activation. For full activation of T cell, we need secondary signal. If this secondary signal, listen carefully, if this secondary signal not there, T cell not active. Understand? If this secondary signal not there, T cell not active. Can kill this virus cell or any bacteria? No, because T cell not active. Understand? T cell not active. So for T cell activation, for T cell full activation, I need what? For T cell full activation, I need what? I need this signal. Understand? So what is energy? What is energy? No secondary signal. Understand? Energy mean no activation, no full, better word here, no full activation of T cell. This is energy. Understand? This is the energy. Let's see what happened here. Stop the co-stimulation if it's an energy condition, okay? Stop the co-stimulation, the stop of second signal, stopping of second signal. How we stop here? Okay, let's see. We having this protein, keep in mind, CTLA4, cytotoxic T lymphocyte antigen, cytotoxic T CTLA4, antigen number 4, okay? This antigen present on T cell. Normally, see here, B7 bind with CD28. This you know.
8.
Okay. If this B7 bind with CTLA4, if B7 bind with CD28, cell become active, okay, because a secondary signal there, yeah. So if B7 bind with CTLA4, no activation, stopping of second signal, understand? So replacement of CD28 by CTLA4. replacement of CD28 by CTL shall not become fully active. Shall not become fully active. Clear? So CTLA4 doing what? Inhibit the, you can see here clearly, inhibit the T cell activation. Inhibit the T cell activation. Clear? When T cell become active, T cell produce what? T cell produce interleukin 2. Interleukin 2 act on same cell, also on other cell become fully active. This is third signal. The second, third, it's a tertiary signal. Okay. So when the CTLA4 there, CTLA4 block the production of interleukin 2, no more further activation of T cell. Clear? This signal, okay, let's see clearly. When T cell active, antigen come from outside, T cell active, T cell start fighting, done. Okay, when fighting done, everything is okay. Now we need to stop activation. Now we need to stop activation. How we stop activation? T cell express CTLA4 on their surface. Then this CTLA4 combined with B7. Then see that CD4 cell or CD8 cell, that cell become inactive. Because when the job is done, of course we also need inhibition there. Okay, let's see here more clearly. When you need defense, T cell active. You can see here how active. B7 with CD28. T cell become active. Killing done. Everything done. Killing done. Everything done. Job done. Bacteria killed. Okay. Virus killed. Virus infected cell killed. Everything done. When the job is done, then we need to inhibit that cell. You must give to order. You must give order. Oh, CD4 cell give order, please. Everything is job is done. Bacteria is dead. Outsider is already killed. Please go to rest. Okay, go to rest. What is the rest? Inhibition. So this we also need. Understand? First activate T cell, then inactivate T cell. First activate T cell, then inactivate T cell. Clear or not? Okay. First activate T cell, then if we just imagine you don't have, your body cell not have CTLA4. What happened with you? If you not have this, what happened with inside your body? Your T cell become active. Okay, active done. Can they go to rest again? Can you inhibit them? No, you can't inhibit them. Okay, you can't inhibit them. If you can't inhibit, then what happens? They start fighting with our body cell. They start autoimmune disease. Oh, it means this is also very important for us. First I make T cell active, then T cell must rest. T cell must be inactive. Clear? So if you not have this, so T cell become active, no inhibitory signal. Clear or not? No inhibitory signal. So there is a balance between activation and inactivation. Clear? Balance between activation and inactivation. Okay, we just imagine another clinical scenario. If your body have lot of CD4 cell or a T cell have lot of CTLA4, they not have CD8, they not have CD28. If they not have CD28, just have CTLA4, what happens? T cell only inhibited, they're not activated. If they're not activated, oh, my immune system in energy. My immune system receive antigen, but only little activation there, not full activation there. They're not able to kill the virus and bacteria. I mean my immune system in energy state. What is that state? Energy state. Energy mean my immune system not fully active. Understand? So still infection there. Bacteria not dead. Oh, not dead yet. Immune system still, no bacteria is there, but they're not killing them. Why? Because the T cell not fully active. Understand? So there must be balance between activation and inhibition. There must be balance between activation and inhibition. Now let's see what are the some clinical points about CTLA4. Okay, the mutant T cell, if they're defective T cell. Okay, if they not have CTLA4, what happens? Mutant T cell, they are not good cell, they not have CTLA4. They can you deactivate them? No, Never. What happened? Autoimmune reaction. Oh, it's also not good thing. It's also not good thing. There must be CTLA4. First CD28 activation done, then inhibition done by CTLA4. So if CTLA4 is absent, then no deactivation. No deactivation mean full activation. Full activation mean autoimmune disease. Clear? They lead to autoimmune disease. Okay, another clinical use you see here, Abtacept. This is a drug, okay? It's a drug. It's immunoglobulin. It's immunoglobulin. What this drug is doing? What this drug is doing? Increase CTLA4 level. Increase CTLA4 level, okay? So this drug is very helpful in rheumatoid arthritis. What is rheumatoid arthritis? Rheumatoid arthritis is autoimmune disease, okay? Rheumatoid arthritis is autoimmune disease. So what happened in this disease? In the rheumatoid arthritis, T cell very active, okay? T cell too much active. What you need here? I need to inhibit T cell. I want to inhibit T cell. Clear? I want to inhibit T cell. So this drug doing what? This drug increase CTLA4. So when you increase CTLA4, no chance for CD28 to bind with B7. Okay, then CTLA4 bind with B7. If these two combine, what happens with the T cell? T cell inhibit. Oh, this drug is very good for rheumatoid arthritis. It's the best drug, but it's expensive. Abtacept. Abtacept is CTLA4. It increase the level of CTLA4. Clear? So this is also some useful condition about the CTLA4. Next, administration of antibody against CTLA4 also enhance your immune system against the cancer cell. Oh, it's also another clinical use here. So if you destroy, listen, if you destroy CTLA4, then what happens? Immune system become more active against cancer cell. If you destroy CTLA4, immune system become more active against cancer cells. So it's also useful. So antibody against CTLA4, this antibody help us to remove some cancer cell from our body. These are the three clinical uses, okay? They are clinical uses of CTLA4. Clear? Some clinical application here. CTLA4, if our body cell not have CTLA4, autoimmune disease there, okay? If you increase the CTLA4, like using medication Abtacept is also helpful. This CTLA4 will helpful in case of rheumatoid arthritis, okay? The CTLA4, if I use antibody against CTLA4, application of this antibody, so will help to increase our immune system against cancer cell. These three clinical application, okay? This concept you clear about CTLA4. So what is CTLA4? Normally, normal physiology here, normal function of CTLA4 after the activation of T cell, give inhibitory signal to T cell, okay? When the job is done, there must be inhibition. So there must be balance between activation and inhibition of the T cell. If overactivated, if totally inhibited, so it's not a good situation, okay? It's not a good situation, clear? How the T cell recognize the antigen, okay? Antigen, how helper T cell that receive antigen, how T cell recognize the antigen? Two way. They can receive antigen, the T cell, they receive antigen by MSC1 and they receive antigen by using MSC2. There are two pathways, okay? Some endogenously synthesized antigen and some some exogenous pathway. The two pathway of antigen, okay? There are two pathway of antigen. The T cell receive antigen from the cytotoxic T cell. What are the names of cytotoxic T cell? These are CD8. What are the helper T cell? Helper T cell, CD4. Helper T cell, CD4. Cytotoxic T cell, CD8. They receive antigen by MSC1. Helper T cell receive antigen by MHC2. Okay. MHC1. Virus inside the cell. Oh, it's a virus inside the cell. Virus already inside the cell. If virus already inside the cell, how the like outside we having here CD8, okay? Virus, this a virus, maybe HIV virus, maybe virus like hepatitis virus, HCV virus, HPV, any virus, okay? Cytomegalovirus. So you know virus attack, how attack the body cell? Virus enter into cell. This CD8 cell not 4. This CD8 cell is passing close to cell. Can recognize? No. So I need the signal here. I need here one signal here. What signal here? So this MSC protein, rough endoplasmic reticulum, okay, produce some proteins, okay. Golgi proteins pack that protein, Golgi bodies, yeah. So then they produce MSC1 protein. This MSC1 protein coming out of the membrane, and take this antigen along with. Understand what the meaning of endogenous synthesized antigen? It's a endogenously MHC1. If the antigen already entered to body cell, okay, then MSC1 protein come on the surface. For example, this is the liver cell, okay, this is the liver cell. Then this MSC1 take this antigen outside and showing here. Oh, now CD8 cell, now, oh, this is antigen here. Then recognize and kill the full cell. Kill this liver cell completely, because the liver cell, I just give you one example there, okay. For example, when the cell in danger, if the some thief enter your home, you're alone at home, how you give signal to the to neighbor? Maybe you shout, maybe you give some green signal for help. The same situation like this. When viruses enter the body cell, they call for help, okay. The bodies, because virus is not a good thing for them, because the body cell, no, I've been killed by this virus at one time. When virus become full active, that cell dead. So that cell, the liver, I just give an example here, liver cell, just a raised hand. The CD8 cell every time circulate in body, okay. Patrolling of police at the night time, see if there any security issue in the full city is in peace or not, okay. So same thing here. CD8 cell is circulating in 24 hours. 2-3 times the CD8 cell is circulating in our body, okay. And what the CD8 cell doing here? The CD8 cell just searching for any cell have some MSC1 protein. Normally MSC1 protein present inside the cell, produced inside the cell, okay. So when the cell have the virus, that virus taken up by the MSC1, and then MSC1 coming out. Oh, it's mean this cell have another cell, neighboring cell not have this. Neighboring cell not have any virus here, but this cell have virus. This cell doing what? Produce MSC1 and take this antigen to outside. Then this CD8 cell can see easily, oh, this cell is infected. Then CD8 cell capture this cell and kill the full cell. Virus infect. Otherwise, what happened? When this cell dead, all the viruses release out and infect other. So CD8 cell kill the virus completely, okay, and kill that cell, kill virus infected cell. Understand this concept clear or not? What's mean endogenous way? MSC1 protein produced inside the cell and capture the antigen from inside the cell, mostly virus infected cell, okay. So these antigen viral in nature, and these antigen they presented on the surface. Of cell, and then recognized by which cell? It's MSC1 recognized by CD8. CD8 recognize the virus infected cell and kill that cell. This is the endogenous way. Clear? So what is the exogenous way? Exogenous way is done by MSC2. For example, what's the exogenous way? It's a macrophage, okay. This is the macrophage. Macrophage fighting with bacteria. Okay. Macrophage fighting with bacteria. For example, you fighting with your enemy, and your enemy a bit stronger than you, and then you have when you're fighting with the enemy, you have some particle of that enemy, some pieces of cloth, yeah, you're fighting. Then you call your big brother, please help me, I want your help. Who's their enemy? I have some clue about that enemy. So just showing that clue is the antigen, this bacterial product, this macrophage and phagocyte this bacteria and try to kill that bacteria and break in many pieces. Bacteria broken down in many pieces, and these pieces now presented on the surface. How they presented? They presented on the surface with the help of MSC2. They presented on the surface with the help of MSC2. Clear? So this MSC2, so this cell, what is this cell? Antigen presenting cell. Clear? Antigen presenting cell. This antigen given to which cell? Antigen given to CD4 cell. Done? This is the way. Two protein, they're different, they work differently. Okay, so MSC2 exogenous antigen presented to CD4 cell. Endogenously synthesized antigen, they presented with the help of MSC1, with the help of MSC1 given to CD8 cell. Clear? The two different way. MSC gene, each body, every body person, their MSC genes are not same. Okay, we having two group. For class 1, we having 3 genes actually: human leukocyte, human, H for human, L for leukocyte, A for antigen. Okay, human leukocyte antigen A, human leukocyte antigen B, human leukocyte antigen C. We having 3 gene for class 1. For class 1 MSC, we having 3 gene. Okay, for class 2, we having DP, DQ, DR, mainly these 3. So nowadays we also having this DM, is not much special. Okay, DP, DQ, DR. We have further category DP1, DQR, DR, DR1, 2, 3, 4, 5, like this. Too many families. For this is very complex. Class 2 is more complex than class 1. Okay, so these are the genes, MSC gene present in our, like every person may have HLA1, have any person may have HLA DPU, another person, your sibling have HLA DQ. So all the person, human leukocyte HLA antigen not same. So when we doing the transplantation, at that time, we not just match the blood group. Okay, we also need to match what important thing, we need to match HLA gene. HLA match. Okay, HLA match. If their HLA gene are same, then very good for suitable, that's very good condition for transplantation. Okay, if they are the donor and recipient, their HLA totally different, the big issue for rejection. This is in the case of rejection there. Okay, so then what happened? There will be rejection there. Okay, let's see about the MSC1, or the structure of MSC1. MSC1 having alpha chains and beta 2 microglobulin. The 3 alpha chain, alpha 3 here, alpha 2 here, alpha 1 here. The 3 alpha. Okay, 1, it's a 2, it's a 3. 3 alpha chain, and along with alpha chain we having beta 2 microglobulin. This one. This is the structure of MSC1. Structure of MSC1. And this area for binding is a peptide binding group, which means it's the antigen binding site. Okay, it's the antigen binding site. Here is the antigen binding here. So this is a structure for MHC1. Okay, structure done. Next, see, structure done. What is the location of MSC? MSC1 present where? All body cell. Every cell of the body having MHC1 except RBC, because they not have nucleus. Okay, all nucleated cell, like liver cell, kidney cell, lung cell, intestine cell, skin cell, brain cell. So every body cell, if they have nucleus, they having MSC1. Clear? They having MSC1. So how they present? Some endogenous antigen. Okay, the synthesized antigen MS, a big example here, some viral protein. Clear? So every body cell, it's the liver, all liver cell they having MHC1, all liver cell they having MSC1. Okay, the MSC1, this MSC1 protein produced inside the cell. All liver cell they have ability to express MSC1. Understand or not? All liver cell, all body cell, not just liver cell. So at what time? The question here, what time cell? Listen carefully. I asked you a question here. What time every liver cell, every kidney cell, all body cell have ability to produce MSC1? I want to ask here, what time cell express MSC1 at every time? No, not at every time. What time? When the cell is infected by virus. If the cell has something wrong inside, or if the cell has something wrong inside, some tumor like changes inside, if the cell become tumor, or cell the in future change to cancer, tumor cell, they express MSC1. If the cell change to cancer, they express MSC1. Then this MSC1 detected by which cell? Which cell is every time circulating? CD8 moving here and there, and just looking for what? Looking for MSC1. MSC1 just like a tag. It's just like a tag. If any cell have MSC1, that CD8 cell recognize that cell and kill that cell. Okay, so this is the reason. Not every time, every cell not express every time. Understand? So when you have something wrong inside the cell, liver cell have hepatitis A, E, B, C, any viruses there. Okay, any cell have some viruses inside them. So what happen? That cell start producing MSC1 to show which cell, just to express I have virus inside me. Cell want to show I have virus inside me. So then MSC1 protein come to the surface. Not all time on the surface. Okay. So at what time express? When cell have something wrong. Any body cell have something wrong, and then they showing some like red flag. Oh, I am in danger. Just the raising hand, I am in danger. So what is the red flag? Oh, it's a red flag is MSC1 protein. It's MSC1 protein is a red flag. So MSC1 protein, clear? So MSC1 protein. Now this flag, CD8 cell easily can identify. Can cell go into cell to find out which cell have virus? Virus living inside the cell. How cell CD8 cell know this cell have virus or not? How no? If the cell express MSC1, then CD8 cell easily identify, oh, you have the virus. Then CD8 cell capture that cell and kill that cell. Understand? So what's the MSC1 role? Everyone clear about MSC1? How is working? How is working?
Sir.
Okay. So if the cell have something wrong inside, so that cell just raise hand. Raise hand. That raised hand, like you have the illness. Who are ill? Who are ill from you, from the class? Sir, I am ill. I not ran the class yesterday like this. Oh, you have some problem. It's like this. CD8 cells circulating everywhere and asking and looking for and looking everywhere. It's just searching everywhere. Any cell infected, any cell infected, any cell infected. Which cell is labeled with MSC1? MSC1 come on surface, yeah? Expressed on surface, having antigen. Expressed on surface, having antigen. Now CD8 cell easily identified that cell and kill that cell. That's about MSC1. This is how MSC1 is working. Okay, so what is the location, what is the structure of MSC1, what is the location of MSC1, what is the way of MSC1 for presentation? There are three things we've done. Okay, and it's all about MSC1. So what are the gene of MSC1? HLA-A, HLA-B, HLA-C. Human leukocyte antigen A, human leukocyte antigen B, these are three genes for MSC1. The structure we having alpha chain, alpha 1, alpha 2, alpha 3, and one beta 2 chains. Okay, so this is the structure here, and how they present the antigen. They're using endogenous way. They're using endogenous way. MSC1 protein used by virus infected cell and used by tumor infected cell. Okay, the tumor, if they're having some tumor like condition inside them, have some abnormal protein, so they express MSC1. Clear? Those they express MSC1. And what is the location of MSC1? MSC1 present on all body nucleated cells. So every body cell have MSC1. Okay, every body cell have, but not every time express. This is very important thing. Okay, every body cell have ability to express, but only shown in which condition? When they have something wrong. Okay, how about the class 2? Class 2. What is the location of class 2? Class 2 present on APC cell only. Understand? Class 2 present on AP cell, antigen presenting cells. What are the antigen presenting cells? Macrophage. Antigen presenting cells, B cell. Antigen presenting cell, dendritic cell.
Dendritic
These
cell.
are most important. Someone also saying Langerhans cell inside the skin. They also working like antigen presenting cell. But you guys must keep in mind these three important for us. Okay. When they found any antigen like fungal antigen, antigen like bacterial antigen, so these things attack from outside. They not go into the cell. These things attack from outside. So they captured by the macrophage are directly antigen going to B cell, directly antigen going to dendritic cell. So what they're doing, they process that antigen first. They process that antigen, okay, like they make what, endosome and phagocytosis first, okay. Then they express with protein. Which protein? MHC2. Which protein on surface? MHC2. Which protein on surface? MHC2. Clear? Then they're moving where? Do you know the difference here? Which one is big leader? CD4 or CD8? One thing about CD4, CD8, which one is more bigger? Like according to the rank or according to the function, which one is more bigger? CD4 or CD8?
CD8.
Now CD4 or CD8, which one is